Hi, everyone. Yeah, my name is Kaspar, Kaspar Podgorski. I am the head of the optical physiology program at a new Allen Institute called the Allen Institute for Neural Dynamics here in Seattle. Everything that I'm going to show you today was done while I was at the Genelia Research Campus, me and my team, but there's also a lot of collaborators involved from all over the place. I'll do my best to call out those folks, and certainly I'll call everyone out at the end. So I'm going to talk today about work that my lab has been doing to establish methods for recording large numbers of synaptic inputs from individual neurons, especially in vivo. Oh, this movie wasn't playing. That means both indicators and microscopes towards this goal. So this talk's going to be pretty informal. I'm going to try and take a page from Darcy. If I can channel Darcy, I'll feel very successful. Please feel free to interrupt me and ask questions and yell and scream and whatever you like. Really, I'd love for this to be interactive. Okay, so much like many of you, my lab develops tools for neuroscience, and specifically, we work on developing both microscopes and fluorescent indicators so that we can do new experiments in neuroscience. And by way of introduction, this fits into a very long tradition that might have started with the work of Santiago Ramon y Cajal, who used Ernst Abbe's first corrected microscopes in combination with Golgi silver stains to do a lot of really cool work on the anatomy of the nervous system. In our case, my lab is working on both microscopes and dyes indicators to do new types of functional imaging. So already yesterday in the workshop, we talked a lot about calcium imaging, as well as a little bit about somatic voltage imaging. And those are both great ways to measure the output of neurons, basically spikes. So for example, this is calcium imaging of neurons in the hippocampus and CA1. A lot of these are place cells, meaning that they fire when this mouse is in a particular place in its environment. A thought experiment that I want you to go through is, what if you want to understand not just when a neuron fires, right? So like its place field, the particular locations in space, but what computation it's performing, right? So how is it computing that place field? What inputs is it receiving that allow it to produce that output? And how does it combine them in order to produce that output? What information does it receive? And what transformation does it perform? So those are the sorts of questions that my lab is working towards developing tools for answering. I feel that traditional, like cellular resolution calcium imaging is probably not the best way to answer those questions. And so the approach that we're taking is instead to try to find ways to directly measure the inputs to a neuron and its output so that we can infer the transformations from inputs to outputs, the computation that a given neuron performs. So this is a different view of those same cells, CA1 pyramidal neurons. And the point of this picture is just to show you that each neuron is an incredibly complex machine. So neurons perform computations by receiving inputs at their synapses that lie throughout their dendrites, as well as on their soma. And it combines all those many inputs, thousands of inputs, into an output. Those inputs come in via chemical neurotransmitters. And as a big part of what we'll talk about today is about 80% of the synapses in the mammalian brain are glutamatergic. They use the neurotransmitter glutamate. A cool sort of anatomical thing is that a lot of those glutamatergic synapses come onto these little protrusions called spines that you can see here. And so you can see just how many synapses each of these neurons has. There's a relatively long history now of measuring synaptic activity in spines using calcium indicators like GCAMP. So these are some beautiful recordings in mouse visual cortex where a mouse is being shown different directions of motion, and there are little flashing calcium influxes into these spines that are caused by NMDA receptor opening. So there are receptors that are calcium permeable that under certain conditions open and allow you to see inputs into individual spines. But the problem with using calcium imaging to record synaptic inputs is that in the vast majority of neurons, back-propagating action potentials, which also cause a flood of calcium into cells, completely swamp the spine-specific signals. So this is a video from a very elegant and thorough study by Aaron Kurlan, Boaz Mohar, and their colleagues. And what you're seeing here is this red circle highlights a particularly independent spine where you can sort of see little bits of input that are coming into that spine. But you also see the vast flashes of calcium that come into the cell when the cell body, the thing that we were measuring in those other experiments, the spiking of the cell, causes the whole cell to light up. And so that is a huge confound, right? Because sure, you can censor the cases when the cell spikes, but if the input from the synapse affects the cell spiking, then you're censoring exactly the situations that you care about studying, right? Which is how the inputs are combined to drive the cell spiking. There's a lot of nonlinearities here, right? Where calcium diffuses inside cells. The opening of NMDA channels depends on the postsynaptic potential. And so if there are other inputs coming in, that's going to affect the amount of calcium that comes in. I mean, there's a whole host of these nonlinearities. NMDA currents themselves are nonlinear. The calcium indicator is nonlinear. People have worked on addressing this to sort of to be able to isolate the inputs to individual synapses, but the problem is really quite fraught. And so, you really have to think of calcium as this highly transformed postsynaptic signal that does, in some cases, reflect spine inputs. In the case of non-spiny synapses onto the soma or onto dendritic shafts, I think the problem is much more difficult even. Yeah, I mean, we can just keep talking about this. Sorry, I'm ranting. But it's a really challenging problem, especially if you want to measure, say, whether two inputs close together are correlated, because they're going to be correlated by all of these postsynaptic processes. Okay, so like that host of motivations and the challenges associated with calcium imaging of synapses led was a big motivation for developing glutamate indicators. To have a better way, basically, of measuring synaptic inputs. And there's basically just one family of popular glutamate indicators, and those are called iGluSniffer, which is the intensiometric glutamate sensing recorder. And that was developed by Lauren's Luger lab first in 2013, and then some improvements were made by Lauren's lab in 2018. There's some other important work by other labs as well on these indicators. So iGluSniffer, it actually has a structure that's very similar to GCAMP, right? Where there's a barrel-shaped fluorescent protein that in the absence of glutamate is dim. There's a domain that binds glutamate. That domain happens to associate, that's connected to the fluorescent protein. That domain's actually split. The importance of this smaller second domain is not exactly the same as in GCAMP, but we don't have to go into the details of that. When it binds glutamate, there's a conformational change in this glutamate binding protein that then makes the fluorescent protein brighter. Okay, great. This particular glutamate binding protein, GLUT-I, actually, wow, this is a typo. Sorry, GLUT-I it was selected because it looks like a Pac-Man, basically. It undergoes this like really big conformational change. There's a hinge that closes when the protein binds glutamate. So here's a structural model of GluSniffer, right? And so this green here is the fluorescent protein, and you can see the chromophore here, and it pokes out. And much like in GCAMP, there is basically an opening in the barrel of this fluorescent protein that where the chromophore actually is accessible to solvent, and basically the movement of these domains here causes some changes in the structure of this fluorescent protein that affects its brightness. What you're looking at here is a soluble protein. So we can express something that looks like this. And if you expressed it, it would be dissolved inside the cell and it would be everywhere. The glutamate concentration inside cells is rather high because obviously glutamate is an important metabolite. It's an essential amino acid. But what you want to do is you want to take something like this and then display it on the surface of a cell in order to measure the glutamate that's actually impinging onto its surface. And of course, the team that originally made GluSniffer did that. So they fused it to a transmembrane domain. And that transmembrane domain, I'll talk a little bit about it later. It's derived from the transmembrane domain of a growth factor receptor called PDGFR. It expresses quite nicely on the surface of cells. And you can see when you when you add glutamate, the indicator on the outside gets brighter. Of course, the indicator that's stuck on the inside of the cell is already in its bright state. Whereas the stuff that's on the outside then goes into the dark state because there isn't a lot of glutamate. And then when you add glutamate, it gets bright. Okay. So it also does that on neurons. And so on the left, here's this first generation paper even about the first generation of iGluSniffer. And here, this is showing glutamate uncaging in a slice where glutamate's uncaged at this yellow spot here. The authors imaged this spine, and they also patched the neurons so they could measure the EPSP associated with glutamate uncaging and also measure the fluorescence. And under these conditions, this indicator looks quite nice. It responds on a not too dissimilar time course and with a not too dissimilar amplitude. But of course, glutamate uncaging is not the same thing as synaptic release, and we'll get to that. So they also imaged, for example, does someone have a question? Oh, no. So they also imaged on individual spines in, for example, here in the visual cortex of a ferret. And so it's kind of cool, they show the ferret different directions of motion stimuli. And because the presynaptic neuron has a preferred tuning to those motion stimuli, it likes particular orientations. This is well known about the ferret visual cortex. They see larger signals to some orientations, regardless of the direction of motion, than others. But there's already a bit of a problem here, right? There's already something weird about the traces on the right, which is, notice that even though this spine is responding, so here are the averages, responding to certain orientations more than others, it basically has a response to every stimulus. And we know from recording spikes from neurons in the ferret that most of them are very deeply tuned, right? So they really basically are supposed to fire no spikes to the non-preferred directions. Of course, this is just one example spine, but I'm going to come back to this issue in a moment. So when I started at Janelia in 2015, I set out to use this glutamate sensor or a variant of this glutamate sensor to map synaptic inputs onto dendrites. And in these experiments, we basically did the same thing as in the ferret. We showed mice moving grading stimuli of different directions while imaging actually two indicators in this experiment. So a yellow glutamate indicator, which is shown here in cyan, as well as a cytosolic calcium indicator in order to sort of compare and contrast the signals coming from each. You image a bunch of spines at the same time. Different spines on the dendrite have different preferred stimuli, which is kind of cool. And you can plot that in different colors, right? In these images, basically the hue corresponds to the preferred direction of the signal, right? Meaning that, for example, red means that those particular pixels in this image responded most to sort of these directions, close to 0 degrees, much more than to other directions. And the intensity of each pixel corresponds to how strong the response is. So everywhere where it's not black, it means there's a response. The image on the left here is the glutamate signal, and the image on the right here is the calcium signal. This calcium indicator, in general, something that everyone should know is that the red calcium indicators are not very good. And so, there's a bit of noise and kind of difficult to see what's going on here. But, there are spines that light up in this calcium signal. And in general, there's very little response in the dendrites. But that's not true of the glutamate indicator. And that's a little bit odd, right? Because everywhere that you see intensity, it means that there's a response. And so for example, in this region of the shaft here, it means that there was actually quite a large response to the stimulus. And we see these large responses basically everywhere, even where there's no spine. And what's a little more worrisome even is this sort of gray color that the shafts often have, which means that they're quite weakly tuned, which might mean that they're a mixture of signals from multiple presynaptic neurons. If anyone has questions, please interrupt me. Another way to like sort of quantify that concept, right, of whether basically there are signals spreading to where they shouldn't be, is to calculate the autocorrelation of those tuning curves. If you measure pixel-wise tuning curves, you can measure their autocorrelation in space, which gives you then a sense of the spatial extent that the indicator samples in brain tissue. And you can do this sort of in a reliable way that it's not affected by, for example, movement, and where you expect it to decay to zero under most conditions, except due to the sort of the structure of the tuning response. And so here you see these spatial extents, and the spatial extent of this particular igloo sniffer variant is multiple microns. And there are different variants of igloo sniffer. This variant is an A184S variant, and those are particularly bad. But even the best variant actually has a spatial extent of about 1.5 microns.
I have a question.
Yeah.
So I don't do these experiments at all, but it seems like this would work better if you knew what the presynaptic partners were. Like one spine could be receiving input from multiple presynaptic neurons, multiple types of presynaptic neurons, right? Is this being done in a circuit where you know exactly what the presynaptic input is?
That's right. So spines typically receive one or two inputs from one or two presynaptic glutamatergic neurons, and generally one. But there's a very wide class, for any given postsynaptic neuron. If you go into like a mouse's brain or into the cortex, for a given postsynaptic neuron, there are many, many, many classes of presynaptic neurons that synapse onto them. It's a wide distribution. Most of them are local. Some of them are coming from far away. Being able to identify what those partners are for any given synapse would be extremely valuable. But we don't actually have really good ways to do that yet. It's something that we're really interested in actually at the Allen Institute.
Right, yeah, so I'm just, right now you're just concerned about signals coming into the postsynaptic neuron and not so much as where it's coming from? Is that the reason?
Yeah, so the goal of these experiments is ultimately to be able to understand, catalog, first of all, to catalog the inputs that come into a given postsynaptic neuron. Because neurons are little computers, right? Like they create an output out of inputs, and the output is different. They transform information, right? Information is represented in a certain way across all of the inputs, and a given neuron sort of simplifies that or transforms it into a different representation, right? And that is the core to me of what your brain is doing. It's just doing that many, many times in a large cascade and recurrent structure. And it's very much what artificial neural networks do. And we want to understand how that happens. So that requires being able to measure the synaptic inputs to a neuron with some specificity, right? We want to know what different synapses convey to our postsynaptic neuron. But we also don't want to be sampling like synapses that belong to other neurons. And that's the problem. If we're sampling a volume, say with like diameter of 3 some odd microns, that's going to contain many, many synapses. So electron microscopy, this slide here, shows us that the mean intersynaptic distance is less than 1 micron. There's roughly there's a little bit more than 1 excitatory synapse per cubic micron in the neuropil of the brain, which means that we're sampling with the glue sniffer that I showed you a moment ago, we're sampling something like 30 synapses. And of course, not all of those are going to be active or tuned to our stimuli. And maybe in some cases, it's not going to matter. In some cases, our signals are going to be very specific. But certainly there will be cases where the signal that we measure is a mixture of different axonal release sites, only a fraction of which will actually be connected to the postsynaptic neuron in the sense of fast ionotropic glutamatergic connections, right? So, yeah, I mean, so that's, it's a known fact that synapses signal with incredible spatial specificity, much more spatial specificity than the measured extent of these iGlu-sniffer signals. Glutamate can spill out of synapses, and that does convey very important signals for various processes. But but again, these sort of especially fast ionotropic signals are very, very synapse specific. Okay, so what is the difference then between the mechanisms of that fast ionotropic signaling and the glue-sniffer responses, which aren't spatially specific? And that's something that we asked ourselves in order to do these experiments. How can we basically make a more synapse-specific glutamate indicator? And so, you know, how synapse-specific can a glutamate indicator be? How do we improve on the spatial specificity of the existing indicator? And what tests would you perform to assess that? I think this is worth thinking carefully about. And you know, the answer that we came up with basically is that endogenous synaptic signaling is specific mainly because of two things. The first is the kinetics of glutamate receptors. Glutamate receptors are very fast and they don't saturate. So when a vesicle of glutamate is released into the synapse, the vesicle contains perhaps 100 millimolar glutamate, and that is rapidly on the timescale of microseconds diluted into the volume of the synapse. And then of course it diffuses out, but then it's also taken up by glutamate transporters. So astrocytic and neuronal transporters that actually very rapidly bind and then a little bit more slowly remove glutamate from synapses. So the kinetics is one thing. And the second thing is that those receptors are localized very, very close to sites of release. So actually on a nanoscopic scale even, there are receptors that are specifically localized immediately opposite glutamate release sites. To sort of think a little bit more clearly about that, you can do some biophysical modeling. So if you simulate the release of glutamate, so this axis has been cut here. If you simulate the release of glutamate into a synapse and you assess it very, very close to the site of release, so 50 nanometers away, or farther away, 1 micron away, you get glutamate transits that look like this thing that spikes extremely high for a very short period of time and then decays down, or this slightly slower sort of filtered version of the response, which on slower timescales looks very similar to what you see at the center of the synapse, and only on very, very, very fast timescales does it look different, right? And so you can think about this as the ratio of the synaptic to the extrasynaptic signal at different temporal frequencies. So at low frequencies, the ratio of the two signals is very small. They're very similar, and only at high frequencies is the ratio different, right? And so only basically over short time scales can you distinguish with very high specificity the glutamate signal in one place or another. Okay, so this tells us at least two things, right? Actually, just these two plots tell you that, first of all, in order to be highly specific, an indicator has to be very fast. It has to be able to report the differences between this blue and orange trace, which means that it has to have components of its signal that are on the order of 100 hertz. And the other thing is that the indicator itself has to actually be close to the release site, because this difference actually comes from spatial differences that are on the nanometer scale. So we can then start to list a set of desirable properties for a next generation glutamate sensor. And so I just mentioned, fast and close to release sites for two things. And a slightly more subtle insight came from carefully considering the kinetics of iGlu-sniffer. So the kinetics of iGlu-sniffer, it turns out, can only be described well by a model that has at least 3 states of the indicator. So basically, there's this initial step where the indicator binds glutamate and forms a bound but usually dim state, followed by a rate-limiting, much slower transition to a bright fluorescent state. And a consequence of this model, which we can observe in vitro using stop flow is that as you increase the glutamate concentration and you measure the rate of binding, the rate of fluorescence change, not the maximum fluorescence, but the rate at which the fluorescence changes to these brief impulses of different concentrations of glutamate. As the glutamate concentration gets higher, you get a saturation. It doesn't turn on any faster. And that obviously has big consequences for the signal in synapses, which are at concentrations way out here, beyond 1 millimolar, versus the concentration outside. You want this curve to be very linear, basically, right? Because you want the signal to be proportionally higher inside the synapse versus outside of it. And this saturation can be described by this parameter Km, which is the half saturating glutamate concentration. And you basically want that Km to be much larger than the concentrations of glutamate, even that the indicator even transiently sees when there's synaptic release. So this model, if Km is much larger than the concentration that you're seeing, it's roughly linear. But if it's very small, then it's highly, highly saturating. Okay, it turns out then that Km is something that actually varies between different glue sniffer variants and that we can engineer. So we can measure it and we can change it by several orders of magnitude. So the final quantity that I want to add to this list is a high Km, and it's super important. All right, so I'm going to show you some new variants of iGlue sniffer, which has been sort of a big focus in my lab for the last few years. We engineered these in order to have this property of having a high Km, in order to be very linear with increasing glutamate concentration. And in particular, I'm going to show you the most data on this variant V857, which is the most linear one. So I guess I just showed you that variant has a high Km. Okay, so here are two variants of iGlue sniffer, which we're calling V82 and V857. The V numbers refer to the order in which they were screened in neurons. So V857 is the 857th variant that we screened in neurons. These have a lot of mutations relative to previous versions of iGlue sniffer. In particular, V857 has 15 different mutations, which we've actually reverted each of these mutations individually and shown that they each affect sensor performance. And what's interesting about these mutations is that most of them, so even though these were found largely by randomly mutating this entire protein, we looked everywhere, most of the mutations that we found actually lie at the interface between these two domains of the protein, the barrel, the fluorescent protein barrel that I told you about, and the sort of Pac-Man domain that I told you about, where they touch each other, which is a completely synthetic thing, which was made when this fusion protein was first created, is being refined, basically, as we evolve this indicator to make it better. So V82 has 12 mutations, and then V857 has an additional 3. Or rather, actually, V82 has 13 mutations, and one of them is slightly changed, and 2 others are new in V857. Okay, so we think that the reason why this interface is being mutated is it's actually helping these two domains sort of pack together in a more structured and natural-like way. These indicators perform very well. As purified proteins, they both have a greatly increased fluorescence change in response to glutamate. So this blue, very flat line here that's zoomed in over on the right, that is the response in purified protein of the previous generation of indicators. And we've dramatically increased the response by over an order of magnitude. And then in culture neurons, we similarly get big improvements in performance. So V857 here is dimmer at rest than the parent sensor, but it actually reaches about the same brightness in response to 1 action potential. V82 is actually is brighter than V857, and it has an even stronger response. As you increase the number of action potentials, V857 especially it saturates less, so it continues to increase in its response. Whereas the peak brightness of the parent sensor over different numbers of action potentials doesn't really change, which this is related to that saturation that I mentioned. V857 is faster than the previous sensor, and V82 is a little bit slower. And there are other ways to quantify. So this is the peak brightness quantified here, but then also you can look at SNR in various ways. Okay, so I've shown you that V857 is both fast and has a high Kn. And those properties alone are enough to make an indicator that's extremely sensitive. So glutamate is released from vesicles at synapses in response to action potentials, as I mentioned. But if you block action potentials, you still see what's called spontaneous glutamate release, which happens when basically individual vesicles are asynchronously sort of randomly fusing to the membrane. And IgG-SNFR3 is sensitive enough to easily watch these spontaneous fusion events happening. So you can look through the eyepieces of your microscope and see this stuff happening. And these are called optical minis. So they're related if you know what a mini EPSP is, which you can record electrophysiologically. These are closely related to those. But of course, now we can watch them in space on neurons, which is super cool. So this is a recording in culture. And we've done a lot of manipulations in culture to sort of understand, to basically be able to better manipulate the amount of glutamate that's released, for example, or affect the rate of these minis in ways that have been sort of previously done and measured electrophysiologically. So in this case, we have sucrose added to the culture, which like slightly raises the rate of these minis. But again, you can still see this stuff even without the sucrose. And so you can, just like we saw from Andrea, for example, you can extract from these videos using various algorithms, traces for these different sites. And we use a non-negative matrix factorization based approach, so very similar to something to the stuff that Andrea described. We get these traces for these different sites. And there are many hundreds in a given field of view that we can record simultaneously. And they have interesting sort of weird dynamics of different kinds. And there's lots of structure to these events. So here I've shown you sort of the top 20, and then these are 10 more lower down. This is a typo here, but there's up to 500 sites, and you're still very, very clearly seeing events. So another important property that we had to select for in this screen, we actually selected for a lot of properties at once. I'm not going to go into too much detail in the screening, but another important property is photostability, which we talked about yesterday as well a little bit. So in order to do long physiological experiments and sort of accurately measure different properties of cells, you really need to be able to record for long periods of time. And so this is a 15-minute long continuous spinning disk confocal recording at, I think, 100 hertz, maybe faster than 100 hertz. There's a little bit of bleaching over the course of this recording. Actually, there's sort of a moderate amount of bleaching. But what I want to point out is that the signal to noise ratio actually doesn't decrease at all. It mildly increases in this recording because most of what's bleaching is actually unproductive background fluorescence. And so we were able to do very, very long recordings of this indicator. So with Michael Hoffa's lab at Dartmouth, we performed two what I think are very important experiments to test the linearity and the synaptic specificity of this new IgG-SNFR3 variant. So I'm just going to show one of them here, which is that Michael's lab had noticed a bizarre phenomenon with older IgG-SNFR variants. So A184V refers to this previous generation, a superfolder IgG-SNFR A184V. And so Michael's lab is interested in the mechanisms of synaptic release, and it's really well known that increasing extracellular calcium dramatically increases the number of vesicles that are simultaneously released during an action potential. But when he did that while imaging IgG-SNFR, he got this result, right? There's no increase, which is quite damning evidence of saturation, because we expect that the amount of glutamate released with this change in conditions is roughly threefold increased. And then he did the same experiment with IgG-SNFR3, and now we actually see the increase that we expect from electrophysiological studies. Also, these responses are much larger in amplitude in general compared to the previous indicator, which I already showed you. Okay, the last piece of the puzzle that I haven't talked about yet is the location of the indicator within synapses. Okay, so as part of another study in my lab, we were using expansion microscopy, which is a method where you physically expand tissue. You know, you take it out of an animal, you're doing histology, you physically expand the tissue in order to see things that are smaller than you would be able to otherwise. And we were looking at synapses that were expressing an older version of igloo sniffer, but in this min display, the classic PDGFR-based backbone. In these images, we noticed these conspicuous gaps in indicator labeling right at the postsynaptic density. Of course, there's sort of always gaps in membrane labeling in our experience with expansion microscopy, and they're sort of more or less randomly distributed. But you know, looking at lots and lots of synapses, we sort of noticed this gap, and so this led us to hypothesize that the surface display construct that's used to put glue sniffer on the membrane is actually excluded nanoscopically from postsynapses. And that would explain some physiological data that other labs have collected, and it would explain some differences that we were seeing between responses, for example, on axons versus dendrites. And so we wondered if we could find a construct that actually isn't excluded from these postsynaptic densities, and maybe it would improve the performance of the indicator. So we made a small library, what we're calling a bookshelf, of different membrane display sequences. And then we used that optical mini signal that I told you about a moment ago as the screening criteria. We actually looked at the signal to noise ratio of those optical minis and found variants that actually have larger signal to noise ratios. So this is on a log scale now, have larger signal to noise ratios than that previous display sequence. And we basically found two general classes of these. One is a C-terminal domain from stargazin, which is an anchoring or a co-receptor for AMPA receptors and calcium channels. And the other is this family of GPI anchors, which are another way of sending things to the membrane. And then when we took those winning constructs and did expansion microscopy on them, we discovered that those better performing constructs were indeed better nanoscopically localized to synapses, right? And so this is the original parent. There's this sort of like slight dimming of in these average images of the postsynaptic density relative to the other labeling, and you can just see that there's relative enrichment using this GPI or that stargazin-based backbone.
Sorry, I have another question. This is going to be an extremely dumb question, but why is optical minis a good way to look at the postsynaptic density? Isn't optical minis something that happens on the presynaptic side?
Well, a synapse is very, that's a very insightful question, actually, because when we image these optical minis, we're both axons and dendrites are labeled, right? Mm-hmm. And so we're not going to be as sensitive, for example, to postsynaptic localization as if we had somehow only labeled postsynapses. I think that's a really key point related to your question. But of course, we're labeling both postsynapses and presynapses. So an optical mini happens at a synapse, first of all, where there's both a presynapse and a postsynapse. The glutamate is released from the presynapse, but it floods both the presynapse and the postsynapse. So you can see that signal either by postsynaptically or presynaptically labeling glue sniffer. So we're basically measuring the sum of the pre and the postsynaptic signal, but I guess we saw a change in the postsynaptic signal that was big enough to allow us to select for these better signal sequences.
Okay, so you weren't seeing glutamate inside the postsynapse, like inside the postsynaptic neuron, just at the postsynaptic surface.
All of this is at the postsynaptic surface. All of these signals are at the synapses of neurons, or on the surfaces of neurons. Okay. In general, even if this indicator was inside of the neuron, it's completely saturated when it's inside the neuron. It's just always in its bright state, right? And that's why I showed you that bleaching. That's sort of like that. There's a fair bit of bleaching of the stuff that's always bright inside the neuron, but the stuff on the surface is basically always dark. And that's one reason why it doesn't bleach.
Awesome, thank you.
I have a question also, Kaspar, since you've been already interrupted. So you said that you use NMF to extend the activity. So I'm wondering also, do you identify different type of spatial patterns when there are different type of events? You know what I mean? You know,
there are absolutely different types of spatial patterns to these events. I did a little bit of sorry, do you want to keep talking? Do you have more? Do you want to elaborate more?
You seem to have understood, but I'm really talking about the level of, you know, single spot. If you see sometimes a more diffused, you know, because NMF is based on the fact that you always have the same spatial pattern, right?
Yes. So, okay. So at a given site, there is a characteristic size of event, right? And so if we watch movies like this for long periods of time, there's basically two classes of things. There are things that are, first of all, in focus. So this is wide field imaging, and there's a big problem with measuring the spatial extent of anything in wide field imaging because lots of things are out of focus. But there are very local little flashy events. You know, I guess I'm pointing at one little spot, but there's really hundreds of these spots. And then there are sort of gigantic things, right? And there is real heterogeneity. And even in time, there is a heterogeneity in the amplitudes of events. So if you look at this long photo bleaching trace, there are these rare, huge amplitude events. And so this is not an artifact. This is a real event that has the same time course, but is that much bigger. So there's a lot of heterogeneity here. I don't think that these experiments, even the spinning, so one reason why we did spinning disk confocal is because we were hoping to get rid of the out of focus events so that we could do spatial analysis on the extent of things. But even in this case, it's not ideal because there are still out of focus things that are just sort of correspondingly dimmer. And, you know, we did these experiments very carefully. You know, there's a piezo focus lock on this microscope, but then we still see this sort of heterogeneity of spatial extents of events. And of course, we're very interested in the spatial extent of events because that's sort of what led us into this work in the first place. But in culture, it's really just not, we haven't developed methods that are appropriate for looking at that.
Thank you.
Okay. So, I mean, so we found now localization sequences that put the indicator closer to release sites on average. And so I hope I've shown you that we basically achieved the three things that we set out to do. And so the next step, of course, is to use the indicator and test it in vivo and do some of those same experiments that we originally wanted to do with the older versions of iGlu sniffer. In Munich, Arthur Connerth's lab did some very cool experiments where they performed simultaneous patch clamp and imaging experiments in vivo. So they patched onto neurons and then imaged their axon. So boutons and the axon that belongs to the same neuron, so that when they could listen to the cell spiking, which is what this ephys trace is here, and then image the release that's associated with that spike, right? And so here's a single action potential, and here is the fluorescence trace at a single bouton corresponding to a single action potential. One thing that's very conspicuous is that the adjacent bouton shows no fluorescence trace, and this is these are what failures are. So this is sort of a well-known phenomenon where even though you get calcium influx with an action potential, because vesicle fusion is random, and on average, roughly one vesicle fuses to an action potential, you can get these failures where there's basically no release. So that's very cool. We can see successes and failures of individual action potentials with very high SNR. We compared single AP, known single action potential responses with this iGlu sniffer 3 new indicator, and with what was sort of previously the best available indicator, this A184S indicator. And I think the difference is night and day.
I apologize, Kaspar, maybe I missed it. Was this in culture?
This is in vivo.
In vivo, okay. That's kind of what I thought, but I wanted to make sure. Okay, fantastic.
Yeah, maybe this is not, this is.
Oh yeah, I didn't read the in vivo Lucille. Sorry, my apologies.
Yeah. And we've done a fair bit more that I actually don't have in these slides, I realize, but we've I should say that there's a preprint of this work on bioRxiv now that sort of goes into much more detail on this characterization and these in vivo experiments. And then we went back and did that same experiment that we originally did with this Venus igloo sniffer variant, and we saw this sort of blurry labeling of our responses at dendrites, and the patterns that we see with this new variant are much different. Now, I do think that there are some differences between this variant and, for example, the corresponding green variant, and you should really read our preprint sort of for details on this. But I think it goes without saying that this is a much more spatially specific and sensitive and non-saturating indicator. Darcy?
Yeah, I just have a question. One of the most kind of intriguing things in the original kind of GCamp6 paper was looking at dendrites with GCamp6 in the orientation tuning and looking at the preferred tuning of the neuron versus the inputs. And from what it looked like, right, is that at least looking at calcium imaging on the spines, it looked like even with normal presentations of basically the cellular output looked like a thresholded, slightly non-linearized version of the inputs on the branch of dendrite that they looked at. It looks like that could be the case in here too. So I'm just wondering if you've looked at broad enough swaths of dendrite of the same neuron to see, is it really that the preferred input of the neuron and is because it's dominated by preferred orientation inputs, or is it something more subtle like clustering with dendritic spiking or some other phenomena?
Yeah, so we haven't done that yet. I mean, that's exactly the type of experiment that the indicator was developed to be able to do more conclusively than the calcium imaging experiments. This is going to depend on the neuron, almost certainly. Neurons have like a wide variety of computational mechanisms available to them. I think what we know about visual cortex is that the inputs along any given tuning dimension of tuning, right? I mean, neurons are tuned on many different dimensions, often at a time, right? But the inputs on most of the axes that we've looked at tend to be very salt and pepper random. So the nonlinearities so there do seem to be biases from calcium imaging data that definitely need to be repeated with glutamate imaging. There are some cool experiments where you can isolate feed forward versus feedback input components to neurons, where we've learned a lot about biases in the feed forward input tuning to neurons. But I think that in general, I'm not trying to say that any one method is going to fully answer all our questions about how these neurons compute. But the type of experiment that Darcy alluded to really should be done with glutamate imaging, as he said. And when someone does that, it's going to be very interesting.
The other thing, that's a comment that I think is nice in visual cortex, there's this, for a long time, there was arguments of how many contacts does a single axon make and back and forth. And some of the EM data seems to indicate that at least in some areas of visual cortex, there's many axons that make multiple contacts across a dendrite. And now this is really fast enough from what it looks like to me that with sharp onsets in those peaks, you could pretty much nail down simultaneity pretty well compared to even previous, calcium or any other method right now. I mean, maybe voltage could be better, but the signals are certainly not as high SNR currently. So,
yeah, agreed. Yeah, I think
really beautiful work, Kaspar.
Thanks, Darcy. That means a lot to me for you to say that. So actually, some of the work that I'm most excited about was done in David Kleinfeld's lab at UCSD, where Ray Liu performed a very impressive experiment deep in the somatosensory cortex of mice. And so the experiment basically looks like this. He's interested in measuring differences between patterns of activity on the axons of incoming thalamic neurons. So VPN is this thalamic nucleus that sends feed forward information about whisker activity to the barrel cortex. And then he also labeled layer 4 neurons in the barrel cortex that receive that input as well as a lot of recurrent input. And so then he's then going to compare what the patterns of activity are of the feed forward inputs versus basically all the input that comes onto the dendrites of layer 4 neurons, both feed forward and recurrent, where the vast majority is recurrent. He does this in awake mice, where he's stimulating the whisker at some known rate. And this is all done very deep in cortex, for imaging methods, about 400 microns. And these structures, of course, are much, much smaller than neurons. They're individual synapses. They're basically sub-diffraction in size. Okay, so what does he see? So I'm going to play for you a movie of the activity on the axons as he's phasically stimulating the whisker. So at 5 hertz, he stimulates the whisker, and at 5 hertz, all of the incoming axons activate. And then when he looks at the dendrites, he sees something very different. So now at 5 hertz, he stimulates the whisker, and there's this sort of cacophony, or this, there's this little symphony of activity that's very repeatable from stimulus to stimulus. So there's a sequence of activations, but it spans tens of milliseconds. And that's, the different timings of recurrent input coming back into these neurons that, are really the layer 4 thalamo-recipient, sort of the first layer that receives input from the thalamus, has this incredible amount of recurrent activation. Now, what I really like.
One question, Kaspar.
Yep.
Is there a reason why for this specific experiment they used GPI over PDGFR? Like, was it just random, or did you specifically choose GPI?
Well, so GPI is one of these new variants that, I probably have a slide on that. GPI has better signal to noise ratios for us than PDGFR, because possibly because it is less excluded from the postsynapse.
I see. Okay, cool, thanks.
So what I should mention is that the SGZ variant, which was the most highly localized to synapses, had very poor functional signals in cortex when virally expressed. And it's been a sort of complex mystery. And I mean, the short answer is you probably don't want to use SGZ, but we do have some collaborators that single cell electroporated it. We've also virally infected rat cortical cultures, and they look fine. They look very, very good. In fact, much better than previous generations of igloo sniffer. It's really a bit of a mystery to us exactly what's going on. The anatomy looks fantastic with SGZ virally expressed, but functionally we basically see very low levels of brightness and very poor functional responses.
Would you still say that for axonal imaging, PDGFR is the best option, even though you said that GPI has the lowest signal to noise?
I think that's right. Especially in vivo. So in culture, I would say just use GPI all the time for everything. For axonal imaging in vivo, PDGFR for a given titer of virus is brighter for us. It just expresses more highly at a given viral titer. And that could be for any number of reasons. It could be due to the detailed sequence of the virus. But for dendritic imaging, we did a quite comprehensive study, which might be in these slides. It's not in these slides, but it's in the bioarchive in our preprint. And GPI is, a few tens of % better than PDGFR for dendritic imaging.
Okay, cool, thanks.
Okay. I'm going to speed up. So this is quantification of those previous slides. It sort of tells you about the sort of heterogeneity of the dendritic responses, whereas the axonal responses are very phase locked. The thing that's cool about this is that these are actually intertwined structures, right? And so this really speaks to the spatial specificity of the indicator, right? Because when imaging these axons, of course, all the other axons that create this heterogeneous dendritic response are in the same volume, but we don't see them at all on any of the labeled axons. And so that really tells us that, we have an indicator that has high specificity to the synapses of the structures that we're labeling. And again, the distribution of timing lags is very, very tight for axons, and it's heterogeneous with a spread of tens of milliseconds for spines. Okay, so it's been almost an hour, but I do want to talk to you about microscopy as well. So the recordings that I've shown you are all of like little tiny segments of neurons, dendrites, or axons at any given time. As Darcy alluded to already, there's a lot that we can learn from those types of experiments, and we're super excited to just do, to give this indicator to many labs to do, for example, mapping of dendritic inputs, sort of a little patch at a time on neurons. And even, as Darcy also alluded to, we could look at sort of precise synchrony of different inputs that are onto the same patch of dendrite with the same types of methods that I already showed you. And so, those different, there's like those flavors of experiments are certainly possible and are very important questions for understanding neural computation. But a somewhat loftier goal would be to measure inputs across much broader swaths of a neuron's dendritic arbor all at once. So, the patterns of activity that come into a neuron like this are very structured, and making more measurements at a time, for example, of the distribution of inputs that's coming at any moment to one dendritic branch versus a second dendritic branch, will tell us a lot about how neurons are using, these different, their beautiful structures and different biophysical mechanisms in order to actually implement computations, operations on inputs. But that's an extremely difficult task for existing microscopes. Neurons have many, many thousands of synapses. Even if we're talking about individual branches, we're talking about huge numbers of targets that are distributed in 3 dimensions, are very, very small, require the highest possible resolution, and the signals, even as indicators get better and better, are relatively small, and so we really need the highest possible sensitivity as well. Okay, so to that end, we've been developing a microscope for in vivo synaptic imaging, for imaging 3D arrangements of synapses at very high resolution at very high speed. So that microscope is called SLAP2. Let's see. Oh, yeah, someone has sent the bioRxiv paper to everyone. So that microscope is called SLAP2, and it has some like pretty cool and unique features that I want to talk to you about today. And in particular, it has this unique scan system. Hold on, let me play this. It has a unique scan system that combines a DMD and a line scanner. And that gives us very flexible control over the patterns of the laser excitation that go into the sample at any given time. Which is exactly the sort of thing that we need in order to target all these little synapses. And we've had the first of these microscopes fully assembled and tested for over a year now, but we're still working on software to do these sorts of cool neuroscience experiments and glutamate imaging experiments. So I'm not going to show you glutamate imaging with this microscope, but I do want to talk about the design concepts today. Okay, so these are a few concepts that I want to highlight, and please, I really want this to be a discussion. Feel free to ask questions, right? So what do I mean by a DMD-based scan system, first of all? So DMDs are programmable mirror arrays. They're used in projectors in order to, and they have about as many pixels as your computer screen. And what they are is each pixel is just a mirror that tilts one direction or another in order to keep or reject light that's coming to that pixel. These mirrors can flip at something like 20,000 times per second. And that allows us to very flexibly pattern light. And as opposed to other spatial light modulators like liquid crystal SLMs, they have very, very good extinction, which is also important for getting efficient, being power efficient, which is an important thing in two-photon imaging. However, the tilted mirrors act as a blazed grating, which creates some problematic diffraction effects. So, I mean, I don't want to do like a full review of diffraction gratings, but laser light is spatially coherent, meaning that there are rays that are coming in to each of these mirrors in the array. And then they will only coherently sum, the reflected beams will only sum coherently along directions where the phase of the light coming off of one mirror is going to be the same as the phase coming off the adjacent mirror, right? And that's true if this ray that's bounced off the adjacent mirror is delayed by exactly one wave relative to the center one, or if it's delayed by 2 waves relative to the center one, or 3 waves, or whatever, right? And so that's what the different diffraction orders are. So the 0th order means that these waves are delayed by exactly 0 waves relative to each other, or 1 or 2 or whatever. And the angle of the mirror reinforces the diffraction pattern along a specific direction, which is basically the direction that the mirrors point. And that is called blazing the diffraction grating. And the diffraction grating is only blazed for certain wavelengths. So if you take a particular wavelength of laser light, you might get a very bright spot along a particular one of these diffraction orders. In our case, it's the 3-3 order. So it's the 3rd order on the X and the 3rd order on the Y direction. But then if you take a different color, you're not blazed at all. And it's very important to collect only one of these diffraction orders, because very small changes in path length will make different diffraction orders no longer sum coherently. And so you have to do a lot of work in order to use DMDs with laser light. With that said, we use DMDs with laser light in this microscope because we really want to take advantage of all the power that being able to turn these pixels on and off extremely fast allows us to harness. And so the heart of this microscope is a module that takes laser light coming in, it bounces it off of part of a DMD, and then it re-images. So it passes through a relay. So this is a one-to-one imaging relay with a mirror here. It re-images the bottom of the DMD onto the top of the DMD, where the mirrors are imaged exactly onto each other. So a mirror that's in this bottom, so the center of a mirror in this bottom section gets perfectly imaged onto the center of a mirror in the upper section, and it exactly cancels out the diffraction effects that are created. You basically trace the lines through the ray diagram back out, and everything comes out like it was coming in, essentially. And so that allows us to turn individual pixels on and off, but cancel out the problematic diffraction effects. This only works for efficiency reasons at specific wavelengths. And we had to design this around a specific wavelength. And for other due to other constraints, that wavelength is 1,030 nanometers, which is a wavelength at which we can get extremely powerful lasers, which are also needed to do this style of approach. Because we basically need to put onto this DMD in any given pixel enough laser power that we would be doing 2-photon excitation in the sample with just that one pixel. And then when we're going to turn each pixel on and off, depending on whether or not we want to use it in any given suite. Okay, so this has all been pretty abstract. Let's make it a little more concrete. So the way this microscope works is we generate a line of excitation, which we scan very fast across the DMD. And for any given line scan, so the line scans roughly 20,000 times a second across the DMD. For any given line scan, we're going to have a different pattern of mirrors on in the DMD, right? And so at a given time, let's say if we want to do a raster scan, right? At any given time, we will have a row of pixels open on the DMD. And as this vertical line passes across the DMD, the intersection of those two things is a point. And what ends up happening in the sample is there's a point that travels across the field of view. And then we open one row at a time, and that creates a raster scan. But we have much more flexible control over the patterns on the DMD than just that. So for example, we can scan just subregions of the field of view. And that speeds up the imaging, because now instead of this line of pixels having to traverse the entire height of the DMD, in this region, it's only traversing a smaller number of rows, and we get a proportional increase in speed. And we can do different things in different areas, and the speed up depends on the number of rows that we're going to image in that given column of the DMD. Okay, and that gives us a lot of flexibility and some increased speed relative to other microscope types. note that we don't have to jump between one field of view and another. It's very, very different from using, say, scan mirrors in order to scan one point to another. The real speed up of this approach comes from defining what we call integration regions of interest. And so integration regions of interest are sets of pixels that we expect to fluctuate in brightness as a unit, like the pixels that correspond to a given neuron or a given synapse. And so we've already talked a little bit about how analysis works for calcium imaging data, for example. And what generally happens in analysis of these types of data is that we want to combine photons that are coming from a set of pixels in space. And that's done post hoc in analysis after each of those individual pixels was measured separately. But we can really speed things up by just combining that information at acquisition time by illuminating those pixels at the same time. And so that's a very powerful approach that's available with this method. And so the sequence of DMD patterns that you use to do that kind of imaging looks like this. It's sort of odd, it's sort of kaleidoscopic, but if I pause it, at any given point in time, so the white pixels are the ones that are open, and you'll notice that all the pixels in a given column that belong to a single region of interest are open, but no two regions of interest are open at the same time, right? And so as the laser line scans across this thing, it basically is measuring the intensity of this particular patch in these columns completely separately from anything else. You don't have to worry about signals being mixed between this patch and another patch. It's basically just measuring the sum of all the pixels that you care about in your integration ROI. The microscope cycles through these extremely fast. And now the frame rate for any given region of interest just depends on the number of regions of interest in that column, which is much, much smaller than the number of pixels in that column. So for these 500 regions of interest, they get imaged, every pixel in every region of interest is now getting measured at 3.6 kilohertz in this microscope. And so this is an approach that allows us to measure thousands of objects at thousands of hertz, essentially. Great, and there's no, so whereas like we built a previous microscope that shared a lot of the same concepts as this, but what it did was it measured different regions of interest simultaneously, like it would measure all the regions of interest along a line together. And that microscope required computational unmixing and required very specific calibrations. You had to know exactly where the line was at any given point in time. And so that made it sort of, the processing of the data laborious and time consuming. And with this microscope, you actually can just see the images as fast as you are recording them, because the photons that you collect at any given time, we know exactly where to assign them in the field of view. Okay, so this microscope is sophisticated. It's got a lot of different components, and I sort of want to, I want this to be a discussion. So hopefully I can engage with you folks if you're not feeling it, no worries. But one thing that we need to do with this microscope is scan in 3 dimensions very quickly, because synapses are distributed in 3 dimensions. Now we can change the pattern that we illuminate very fast. So there's no problem with scanning a particular pattern in one plane and then going to the next plane and scanning a completely different pattern the next a few microseconds later. But we need to find a way to essentially move that image of the DMD through the sample very fast. And this is sort of the beautiful classic approach for this is called remote focusing, or it's a particular type of remote focusing, the Bottcher-B-Wilson remote focusing approach. And it basically relies on this idea, right? Which is that if you have an image plane, right? If you're trying to relay a point to an image plane, you can create an intermediate focus and then pick up that focus with another objective, another infinity corrected imaging system that is just sort of slightly displaced. So if you move L3 relative to L2, so let's say you've created a focus, a part of your image with by relaying it from using L1 and L2 to this point here. And now if you move the objective L3 along with the entire system behind it a little bit, you're going to see that point that you created move out of focus, right, on your camera plane. So that seems pretty obvious. It turns out that in the details, the aberrations that are created basically by being out of focus are sort of exactly canceled using the system. The sort of the odd ordered aberrations of the system are canceled. But this system that's drawn in A is actually equivalent to another system that looks like this in B, right? Where you have a mirror at this intermediate image, a mirror here at this intermediate image plane. But now you can simulate moving L3 and this lens and the image plane relative to L2 just by moving this mirror back and forth. And the huge advantage of this is that you can move this tiny mirror instead of your whole objective that's looking at the sample, which is super, super fast. You don't actually have to move anything near your sample. So if you need to scan very fast, you don't have something like vibrating at hundreds of hertz immediately over your sample. It works for the whole field of view at the same time, which is different than deformable mirror-based remote focusing. So we needed this remote focusing system in our microscope, but because we needed to basically have a fixed focal plane on these DMDs that then got remote focused all at once, it turns out that we couldn't use deformable mirrors. Spencer has made very nice remote focusing systems using deformable mirrors, and several other people have here as well. But we were unable to do that, and we had to go with this approach. But we like this approach as well. Another cool thing that you can do, which has been used to great effect, I think to greatest effect with in a slightly different way with single objective light sheet imaging, is that you can actually tilt the image on your sensor relative to the sample. So if you were to tilt this mirror, you could actually sample different tilted planes within the sample. So anyways, this is a very cool approach that I think that everyone, I am very much running out of time. So maybe I will just be super, super quick about a couple other things, just so people are aware of them. This microscope uses silicon photomultipliers. It doesn't use PMTs. And that's because the photon rates involved are very high. And so it turns out that you can, that these other detectors have lower multiplicative noise and therefore perform better at very high photon rates than PMTs. So it's something you should check out if you are working with sort of fast physiological imaging. Certainly any voltage imaging with a laser scanning microscope should be done with these other detectors rather than PMTs. We use polygonal mirror scanners, which are super fast and have some traditional problems, but actually those problems are nicely canceled out with this approach that we use. And one cool thing about using polygonal mirrors is that we actually scan two beams off of the polygonal mirror at the same time, and they have a very well-defined phase relative to each other that is defined by their spatial location on the mirror. So when we're on the center of one mirror, we're exactly on the edge of another one. We also distribute the optical corrections between the objective and the other lenses in the system, and that allows us to have a very high transmission, high NA, low mag objective that performs very nicely. That sort of for an equivalent performance, we get about 10% better transmission across the spectrum. But I won't talk too much about that. Okay, this is a cool microscope. It has some cool features. It's really quite niche in that it has this limitation of 1030 nanometer excitation and a relatively small field of view. But if you're interested in dendritic and synaptic imaging especially, I'd love to talk to you more about it. So thanks very much. We're also hiring at the Allen Institute. Please get in touch if you're interested. I want to thank the huge number of people, this is probably not even complete, that contributed to the development of iGlue Sniffer, as well as a great team of people that have worked on SLAP2, including the team at Vidro Technologies and MBF that have sort of made kits of this microscope available to people and are working on the software. This was a pretty deep collaboration with Leica as well, and that involved our legal counsel at Janelia. So anyways, thanks to everyone. I'm sure I missed some people. Sorry to rush. Thanks again.